Kia ora everyone. So welcome to the final mini lecture that I'm going to give for this course. And what I'm going to do in this final lecture is take our understanding of fungal pathogen outbreaks and apply that to a couple of examples. So we can look and see that we can apply this knowledge to understand something about the nature of fungal pathogens and their outbreaks. So I'll stop my video and we'll get going with the slides. Okay, and so what I want to do is take a look at a couple of examples of population predictions of fungal pathogens and how we can understand those, the nature of those pathogen outbreaks based on phylogeny. So the first example I'm going to look at is a fungal pathogen called Dothostroma septosporum, and this is a pine pathogen that's found across the world. And what we're going to do is we're going to consider isolates that are found in two places, in Europe and in the Southern Hemisphere. And what we want to do is we want to think about where this disease originally comes from. So if one of these two places is the origin of this fungus, and one of them has subsequently escaped from that place and resulted in a new invasion, a new outbreak, what would the genetic diversity of the fungal populations from these two regions look like? And so based on what I told you in the previous mini lecture, you should be able to answer that question. So what do you predict the genetic diversity of the fungal populations from these two regions should look like? And so hopefully you'll be able to come up with an answer for that. But what we're going to do is, if I move to slide number 2, is we'll look and see what the result is when people have actually looked at this question. So what I'm presenting here, this picture, this is a phylogenetic tree of isolates of Dothostroma septosporum that have been collected from across the world. There's quite a lot of detail on here, but there's a couple of things I want you to focus on. The first is that anything that's labeled with an A at the tip, so for example, this top group here, that means it comes from Austria. And this group here in yellow and orange are the isolates from the Southern Hemisphere. And a couple of things to note. So these funny arrow-shaped things, this just means that there's a variety of isolates that are present within here that all form a single monophyletic clade, but we're not looking at the details of their relationships. So we could depict this orange and yellow group also as one of these triangles, but we're just zooming in on these and blowing them up to show the details of the phylogenetic relationships that are not shown for these other groups. And when you're looking at these phylogenetic trees, the trick is not to look down and see how spread out they are in this direction. It's to see how long the branch lengths are in this direction here. And so the more branched the tree is, the more distantly related they are. So based on this, what would you say is the invaded species and which is the origin? And I'll give you a minute to think about that. All right, so what did you come up with? So the answer is, and hopefully you can get this, that the southern hemisphere has been invaded by isolates from the northern hemisphere, in particular from Austria, it seems. So here we have the southern hemisphere group, and they are all one nice little monophyletic clade. And so this is all of Dothostroma septosporum, the diversity of isolates. And so the ones found here only form one part of the clade. Whereas if we look at isolates from Austria, we find them here, 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 spread right throughout the entire diversity of the tree. So it seems like there was one big population, perhaps based in Austria, that contains the genetic diversity of the entire species. But there's only one subpopulation, only one little bit of genetic diversity that's present here in the southern hemisphere. So that suggests that the origin is in Europe, perhaps somewhere around Austria, and then one isolate over here has spread to New Zealand. That's since diversified, but it was originally one isolate that spread to New Zealand in the southern hemisphere. Okay, so hopefully that's clear. Let's move to slide number 3. And now we're looking at the second example, which is the Irish potato famine, Phytophthora infestans. So the Irish potato famine occurred in the mid-1840s and partly resulted from the catastrophic failure of the potato crop in Ireland at times when many people in Ireland depended on that for their nutrition. So it's estimated that about 1 to 1.5 million people died of starvation, and another 1.5 to 2 million people emigrated from Ireland, including coming to Aotearoa. The reason that the potato crop failed is because of a disease called late blight disease, and the causative organism for that is, of course, Phytophthora infestans. Now, Phytophthora infestans is not a fungus, it's an oomycete, but the dynamics are exactly the same. So the question is, where had this devastating disease come from? Why did it suddenly appear in Ireland? And in fact, it occurred around Europe and also North America in the 1840s. So the question we're going to look at here is, what is the origin of Phytophthora infestans? And let's fast forward 150 years. And so what people did was, back in those days, they made collections of infected potatoes. And so now in, say, museum samples, we have access to those original infestations of Phytophthora infestans. So what people did is they took those potatoes and they extracted DNA from them, and they sequenced the DNA of the pathogen from these old historic samples as well as from modern samples. And then they looked at those DNA sequences and they analyzed them using the phylogenetic tree approach that we've talked about here. And so what I'm going to do in the next slide is show you what the phylogenetic tree looks like. And from this, I want you to answer what you conclude. So was P. infestans an indigenous species that spread to this new host, or was there an invasion of P. infestans from elsewhere? So let's go to slide number 5. And here's the phylogenetic tree. So the old samples are indicated in red. The modern samples that are believed to be related to the old samples are in blue, boxed in blue. And then the rest of the ones that are not boxed in either pink or blue other samples from around the world. And once again, so the length of the branches in this direction, in the horizontal direction, not the vertical direction, is proportional to the level of sequence divergence. And so based on this, what would you conclude? Would you conclude that the samples that were present in Europe had jumped across from an indigenous population of Phytophthora that was already there? Or is this more likely to be an invasion from somewhere? And hopefully, if you've understood what I've said, you'll realize that the latter is true. So it's likely to be an invasion, because here there's very little genetic diversity at all between these samples from across Europe. And so it just seems to have been a single invaded population whereas here we have more genetic diversity in the global population of Phytophthora, so the branch lengths are longer in the horizontal direction than they are in the samples that came from the original outbreak. Okay, so moving to slide number 6, the second question that I posed originally that we can now answer is, where did P. infestans come from? So it seems that it's a clonal population, which suggests it's an invasion. And so if it's invaded from somewhere, where has it come from? And I'm not going to show you the data, but more DNA sequence data from Phytophthora infestans from around the world has shown that the most genetically diverse populations are found in Mexico. So this suggests that Mexico is the geographic origin of P. infestans. And so why is that? So if you've been following along with what I've said, that should be obvious why that's the case, but I will go through it. So it comes down to this question of genetic bottlenecking again. So if a species is spread to another area, for example, by a human or by whatever reason, those new areas will only be colonized with a subset of the original population. And therefore the genetic diversity of the colonizing population is necessarily less than that of the original population. So I've got this example down here again of genetic bottlenecking. So here's the original population. Different genetic variants are indicated by different colors. And what's happened is that some of these individuals have been transferred to a new area. So these are in an invasion. And so the genetic diversity of this invaded population is necessarily less than its original host. Now, the numbers of individuals can still end up being the same or greater than the original population. A number of individuals is not the question. The question is how much genetic diversity is there between those individuals. And that genetic diversity in here is still less than that here. And so the regions with the greatest genetic diversity are likely to be where the species originated. Okay, so that's all I'm going to say about that. I'll just finish off by going to slide number 7 and just say that subsequent studies have cast doubt on some of these conclusions about the origins of P. infestans, and they suggest that the origins might be more complex. But you don't need to worry about that. I'm just telling you as a disclaimer to say that, you know, if you're going around telling everybody that P. infestans came from Mexico, that may not actually be true, but we don't have time to go into the details of what these subsequent studies have shown. And so I'm really just using this as an illustration to show how to think about it. And so the key points for this course are so that you can make genetic diversity predictions depending on the nature of the origin of fungal outbreak species, and then also to be able to interpret phylogenetic trees to see which of those predictions end up being the correct ones. And so to tell you something about what the nature and origin of the fungal outbreak was. Okay, so that's it. I'm finishing up now. And as I go to slide number 8, we're just going to go over the learning objectives very briefly to make sure that we've covered everything. So by the end of this lecture, which is actually 4 mini lectures, you should be able to define the phylogenetic terms clade, monophyly, and paraphyly, discuss the phylogenetic relationships of the major groups of fungi, including the placement of the Glomeromycota and the paraphyly of the Zygomycota and Chytridiomycota, describe issues in anamorph and teleomorph taxonomic relationships with respect to clades, monophyly, and paraphyly, and how phylogenetic trees can help with the one fungus, one name paradigm. Predict the differences in population structures of pathogens depending on the nature of the outbreak, and interpret phylogenetic trees of pathogen populations to infer the likely nature and origin of outbreaks. So I think we've covered all that, but again, if you have any questions, please direct them to Piazza, and I will post a quiz on this set of mini lectures on Piazza, on Canvas as well. And good luck for the rest of the course.